Hello everyone, this is the Medical Gateway, and in this part we're going to study regarding red blood cells. We're going to discuss the functions of red blood cells, but before that we'll discuss their numbers and their structure. So red blood cells, in males the total number of red blood cells are 5.3 million per millimeter square, whereas in females the number of the red blood cells are 4.7 million per millimeter square. So number of the RBCs are in a relatively greater quantity in males as compared to females. Now let's discuss its structure. So the structure of the RBC is basically in the form of a biconcave disc. So if you focus on its shape, the shape of the RBC, its diameter is around 7.8 micrometers, whereas its width on the sides, on the borders, is 2.5 micrometers, whereas its width in the center is 1 micrometer. So this difference of width in the center and the peripheries make RBCs a biconcave disc. Now, what is the significance of this structure? Basically, the amount of the membrane of the RBCs is relatively in a greater quantity as compared to the material inside the RBCs. So the material inside the RBCs is less, whereas the membrane outside the RBCs is in greater quantity. So now, what is its significance that having more membrane as compared to the contents inside? This helps RBCs to easily squeeze through any place. If there is a blood vessel or there is a tight junction of blood vessels, RBCs can easily squeeze themselves through them. So this is a biconcave disc. This is how RBC is. What is the shape? This is the shape of the RBC. So now if the RBC has to pass through a blood vessel which has lesser diameter than 7.8 micrometers, then the RBCs will squeeze itself. Now, if this membrane, for instance, in some conditions which we'll discuss afterwards, if the amount of the membrane is reduced, then what will happen? The RBC will not be able to maintain its biconcave disc shape. The RBC will be converted into a spherical shape, and then the contents inside and the membrane both will be in proportion to each other. But what will happen in that case of the spherical RBCs rather than the biconcave RBC disc is that those RBCs will be unable to squeeze through the tight pores, and then the RBCs will be ruptured in that case, as in the case of hereditary spherocytosis, which is a hereditary disorder of RBCs, where the RBCs are converted into a spherical shape because of the less amount of the membrane outside the RBCs. So now let's discuss the functions of RBCs. So RBC has three broad functions. One is oxygen transport, the other one is carbon dioxide transport, and the third one is acid-base buffers. So let's discuss its most important function, which is oxygen transport. So we have in red blood cells, we have a protein, the oxygen carrying protein known as hemoglobin. So hemoglobin is basically an oxygen binding protein which is present inside the red blood cells. So now let's discuss exactly how it carries the oxygen and what is the amount of the oxygen carried in the RBCs by the hemoglobin. For this, we have to calculate the value of the amount of the oxygen carried by the RBCs. For that, first of all, we'll discuss that in 100 ml of blood, there are 45 ml of RBCs. And how we have calculated this, we know that 45% of the blood is basically the cells, and in the cells the RBCs are the predominant cells. So 100 ml of the blood is basically has 45 ml of RBCs. Now and we know that 100 ml of RBCs have 34 grams of hemoglobin. So what is the amount of hemoglobin present in RBCs? So 100 ml of RBCs have 34 grams of hemoglobin. Okay, basically we are going to calculate that what is the amount of oxygen which 100 ml of blood can carry. For that, we first have to know that what is the amount of RBCs in 100 ml of blood, and then we should know that what is the amount of hemoglobin in that 45 ml of RBCs or 100 ml of RBCs. So if 100 ml of RBCs have 34 grams of hemoglobin, then 45 ml of RBCs would have around 15 grams of hemoglobin. Okay, if 100 ml of RBCs have 34 grams of hemoglobin, then what is the amount of hemoglobin in 45 ml of RBCs? We basically want to know that what is the amount of oxygen carried by 100 ml of blood. So for that, first we have calculated the amount of the RBCs present in them. Then we will calculate the amount of hemoglobin present in these RBCs, and then we will calculate the amount of oxygen carried by that amount of hemoglobin. So if 100 ml of RBCs have 34 grams of hemoglobin, then 45 ml of RBCs will have 15 grams of hemoglobin in them. So 100 ml of blood will have 15 grams of hemoglobin. Okay, so in males, what is the concentration of hemoglobin? It is 15 grams per 100 ml of blood in males, and in females it is a little less than 15 grams. Okay, now this hemoglobin has to carry oxygen. So 1 gram of hemoglobin has the ability to carry 1.34 ml of oxygen. So 1 gram of hemoglobin has the ability to carry 1.34 ml of oxygen. So 15 grams of hemoglobin will carry 1.34 multiplied by 15 grams of hemoglobin. So 15 grams of 1 gram of hemoglobin can carry 1.34 ml of oxygen. Then 15 gram of hemoglobin can carry 20 ml of oxygen. If we multiply 15 with 1.34, the answer is around 20. So 15 grams of hemoglobin can carry 20 ml of oxygen. So now we know that 100 ml of blood has 15 grams of hemoglobin, and 15 grams of hemoglobin can carry 20 ml of oxygen. It means that 100 ml of blood can carry 20 ml of oxygen. So this is where everything was leading. Okay, so calculation requires 100 ml of blood requires the amount of the RBCs, then the amount of the hemoglobin in those RBCs, and then the ability of the hemoglobin to carry the oxygen. So finally 100 ml of blood is able to carry 20 ml of oxygen. Okay, now coming to the next function, which is carbon dioxide transport. Now as the blood can carry oxygen, which we have discussed in detail, the blood also carries carbon dioxide from the tissues to the lungs, from where the carbon dioxide is exhaled out. Now what is happening inside the red blood cells is that water reacts with the carbon dioxide, converting it into carbonic acid, H2CO3, carbonic acid. So now the carbon dioxide will travel in the blood in the form of carbonic acid. Now this is happening in the tissues. Now this carbonic acid, when this reaches the lungs, this carbonic acid is converted back into water and carbon dioxide, and this carbon dioxide will be exhaled out. This is how the blood is transporting carbon dioxide. We'll discuss in detail regarding the transport of oxygen and carbon dioxide in the chapters which will come after a few chapters. Now, which enzyme is converting this, is catalyzing these reactions? So the enzyme is carbonic anhydrase. So carbonic anhydrase is the enzyme which converts water and carbon dioxide into carbonic acid, and then back carbonic acid into water and carbon dioxide. Okay, so this is how the carbon dioxide is transported. Then the blood has acid-base buffer system. So basically the proteins present in the blood can act as buffers for the acid-base regulation. So among the proteins, the hemoglobin, which is an oxygen binding protein, the hemoglobin plays the greatest amount of role in buffering of acid base. Okay, so this is also a function of blood. So these are the three main functions of blood: oxygen transport, carbon dioxide transport, and acid base buffers. So this is it. See you in next part.